In this video, we're going to talk about ion movements, like sodium and potassium moving across the cell membrane of the neuron, and we're going to talk about electrical signals that are generated down the axon. To begin, we'll talk about the transmembrane potential. The transmembrane potential, or membrane voltage, is the difference in the electrical potential between the inside and the outside of the neuron. Actually, almost all plasma membranes have an electrical potential across them, with the inside usually more negative with respect to the outside. We're going to talk about the transmembrane potential of the neuron. The neuron has basically three different transmembrane potentials. It has the resting membrane potential, a graded potential, and an action potential. All neural activities begin with a change in the resting potential of a neuron. If a stimulus disturbs the resting membrane potential, then a temporary localized change occurs that's called a graded potential. And if the graded potential is sufficiently large enough, it will produce an action potential. The action potential is then propagated along the axon to one or more synapses at the synaptic terminals. So let's look at the resting membrane potential. In a typical neuron, the extracellular fluid surrounding the cell body of the neuron has a high concentration of sodium and chloride ions, and the intracellular fluid has a high concentration of potassium and negatively charged proteins. The negatively charged proteins can't cross the cell membrane. They're too large, so they stay inside the cell body, thus making the interior of the cell more negative than the outside. In addition, there are leak channels on the dendrites and the cell body that are always open. So there are potassium leak channels, and because of our laws of diffusion, potassium, which is higher on the inside of the cell, will diffuse out to a lower concentration. There are also sodium leak channels, and again, because of the laws of diffusion, sodium, which is higher in concentration outside the cell, will diffuse into the cell towards the lower concentration. Both sodium and potassium carry positive charges, so when potassium diffuses out of the cell, it carries positive charges with it, and when sodium diffuses into the cell, it also carries positive charges with it. It's the concentration gradient that drives potassium out of the cell and sodium into the cell through those leak channels. There are more potassium leak channels, however, than sodium leak channels, so the cell membrane permeability favors potassium, meaning potassium diffuses out faster than sodium can enter. This means that there are more positive charges moving out than there are positive charges diffusing in. Because of this concentration gradient and the leak channels and the negatively charged proteins, the interior of the cell is more negative. Now, this concentration gradient is actually created through passive forces that act across the membrane. And these passive forces are both chemical and electrical in nature. With the chemical gradient, it's that rate of diffusion that I talked about. So because the inside of the cell has a higher concentration of potassium than the outside of the cell, potassium tends to move out of the cell. And because the concentration of sodium is higher outside the cell in the extracellular fluid than it is inside the cell, then sodium is driven into the cell. But there's also an electrical gradient as well as this chemical gradient. Because the cell membrane is more permeable to potassium than it is to sodium, the potassium leaves the cell faster than sodium enters the cell. And then, of course, this causes an excess of negative charges inside the cell due to those negatively charged proteins, and more positive charges outside of the cell because of the positively charged potassium leaving the cell. This leads to a potential difference in electrical charges across the cell membrane. The potential difference is negative 70 millivolts. We know that positive charges are attracted to negative charges. So with the electrical gradient, sodium in the extracellular fluid is attracted to the negative charges inside the cell. This will cause sodium to want to move in faster into the cell. Potassium, which has a positive charge, and we know potassium is higher on the inside of the cell, potassium will more likely want to stay inside the cell because of the negative charges inside the cell. So in order to fully comprehend the movement of ions, we have to look at the electrochemical gradient. The electrochemical gradient is the sum of the chemical and electrical forces that act on the cell membrane. The electrochemical gradients for potassium and sodium are the primary factors affecting the resting potential of most cells. Sodium ions are attracted into the cell by the excess negative charges inside the cell. Sodium also moves by a concentration gradient because it is higher outside the cell than it is inside the cell. So because of this concentration gradient, it also wants to move into the cell. If the cell membrane were freely permeable to sodium, sodium would continue to come into the cell, bringing its positive charges with it until an equilibrium has been reached. This equilibrium potential is positive 66 millivolts, and once the transmembrane potential has reached positive 66 millivolts, there would be no further net movement of sodium. So we could say that sodium reaches its equilibrium potential at positive 66 millivolts. Potassium ions want to move outside the cell because of its concentration gradient, where there are more potassium inside the cell than there are potassium outside the cell. But there's also an electrical gradient with potassium, and the potassium ions are kept inside the cell by the attraction between the positive potassium ion and the negative charges inside the cell. As well as potassium has a repulsion to the outside of the cell because of the positive charges in the extracellular fluid due to the sodium ions. In other words, potassium is repulsed by positive charges. Positive charges repel other positive charges, so potassium doesn't want to move out because of these reasons. However, when we look at the net equilibrium potential for potassium, if the cell membrane were freely permeable to potassium, an equilibrium potential of negative 90 millivolts would be reached, where there would be no net movement of potassium after that. So that means even though potassium is being held inside the cell because it's attracted to the negative charges and because it's repelled by the positive charges outside the cell, there's enough of a concentration gradient between potassium inside and outside the cell, and there are more leak channels in the cell membrane. Potassium will leak out at a fast enough rate, and if it were freely permeable to potassium, then the equilibrium potential would be reached at negative 90 millivolts. But the cell membrane is not freely permeable to potassium, and it's not freely permeable to sodium. There is a continuous leakage of sodium into the cell and potassium out of the cell, and this keeps the resting potential near negative 70 millivolts. Now, any stimulus that increases the membrane permeability to sodium or potassium ions will produce a sudden and dramatic ion movement inside or outside the cell. Now, because of these leak channels, potassium is constantly moving out of the cell and sodium is constantly moving into the cell, there is a sodium-potassium pump that helps to replenish these ions inside and outside the cell. This sodium-potassium pump keeps the resting membrane potential stable. This pump will pump out 3 sodium ions from the inside of the cell because sodium continually is leaking into the cell and we have to pump sodium out. And at the same time, it pumps in 2 potassium ions because potassium is always leaking out, so we have to pump the potassium back in. So the sodium-potassium pump pumps 3 intracellular sodium ions out and 2 extracellular potassium ions in, and this helps to maintain the resting membrane potential at -70 millivolts. The sodium-potassium pump requires energy in the form of ATP in order to move the sodium ions that leak in and to recapture the potassium ions that leak out. So these leak channels are passive channels. They're always open, and they help to create that resting membrane potential. There are other channels, however, that we find on the dendrites and the cell body, and these other channels are more active channels that we call gated channels. Gated channels, like the name implies, they have a door or a gate that opens and closes. So these gated channels open and close in response to specific stimuli. The gated channels can be closed but capable of opening. They can be open or they can be completely closed and not capable of opening. There are 3 classes of gated channels. The first class are chemically regulated channels. The chemically regulated channels will open and close when they bind to specific chemicals. For example, there are receptors on the dendrites in the cell body that bind to a chemical called acetylcholine. And once acetylcholine binds to the receptors, then that gated channel will open. These chemically gated channels are found in abundance on the dendrites and the cell body of a neuron. They're also found at the neuromuscular junction on the motor end plate. There are also voltage-regulated channels that open and close in response to changes in the membrane potential. So that means when the voltage changes, when that transmembrane potential changes, then these voltage-gated channels will open or close. Voltage-gated channels are found in areas of excitable membranes. So that means a membrane that's capable of generating an action potential. So these voltage-gated channels are going to be found on the axon, and the channels are going to be sodium and potassium channels, and they are regulated by voltage changes. So we call them voltage-gated sodium channels or voltage-gated potassium channels. Calcium channels at the synaptic terminals are also voltage-regulated channels. The third classification of gated channels are mechanically gated channels that open and close in response to physical distortion of the dendrites or of the cell membrane surface. So these channels are important with sensory receptors for touch, pressure, vibration, where something is mechanically, physically touching or pressing on the dendrites or the cell membrane. When the cell is at rest, most of these gated channels are closed. The opening of gated channels will change the rate of ion movement across the cell membrane. If sodium channels are open, more sodium will move across the cell membrane. If potassium channels are opened, then more potassium will move across the cell membrane. The voltage-gated channels are restricted to the axons, the branches of axons called the collateral branches, and the synaptic terminals. The mechanically gated channels are typically found on the dendrites of sensory neurons only, and the chemically regulated channels are typically found on the dendrites and the cell body of the neuron. So here in the picture above, we have the resting membrane potential at negative 70 millivolts. And again, the resting membrane potential is created because of the negatively charged proteins inside the cell and because of the electrochemical gradients of potassium and sodium, where the net potassium ions will move out of the cell and sodium will move into the cell. But because there are more potassium channels than there are sodium channels, more positively charged potassium moves out of the cell than positively charged sodium moves into the cell. And those are the things that create the resting membrane potential. In the picture below, we see a graded potential. A graded potential are local potentials, and they are changes in the transmembrane potential that cannot spread very far. So any stimulus that opens a gated channel will produce a graded potential. So on the dendrites and the cell body, there are chemically gated sodium channels. And on these channels are receptors for acetylcholine. So if acetylcholine binds to these receptors, these chemically gated sodium channels open up, and sodium will enter the cell because they're attracted to the negative charges inside the cell and because sodium wants to diffuse from higher concentration to lower concentration. So sodium moves into the cell. As the sodium ions arrive inside the cell, they start to spread out. As these positively charged sodium ions spread out, they start to shift that membrane potential toward 0 millivolts. So the resting membrane potential, which was at negative 70, will become more positive. Negative 69 is more positive. So a graded potential starts at around negative 69 millivolts. As more sodium comes into the cell, that graded potential could become even more positive. Maybe it'll go to negative 68, negative 67, all the way up to negative 61. Whenever the transmembrane potential is shifting from the resting membrane potential towards 0 and above, this is called depolarization. So depolarization is any shift from the resting potential toward and above 0 millivolts. So if the stimulus were to open the sodium-gated channels, then sodium would flow into the cell and depolarization would occur. As the positively charged sodium ions enter into the cell body, a local current in the cytosol will move the positive charges parallel to the inner surface of the cell membrane toward the axon hillock. If the stimulus were to open a potassium-gated channel instead of a sodium-gated channel, then the opposite effect would occur with the transmembrane potential. Potassium would move out of the cell, making the inside of the cell more negative. So from negative 70, if those potassium channels, gated channels, were open, and potassium were to move out of the cell carrying its positive charge, that negative 70 would move to negative 71, negative 75, negative 80, negative 90 millivolts. As the potassium outflow increases and those positive ions are lost inside the cell, we call this hyperpolarization. Hyperpolarization is an increase in negativity of the resting potential. So anytime the transmembrane potential goes below negative 70 millivolts, this is called hyperpolarization. After a cell has gone through depolarization and has become more positive, so negative 69, negative 61, that's called depolarization when the transmembrane potential is above resting membrane potential. In order to get back down to the resting membrane potential, there has to be a process called repolarization. So repolarization is the process of restoring the normal resting potential after depolarization has occurred. Repolarization typically involves the opening of potassium-gated channels. Graded potentials can occur in the membranes of many different cell types. It can occur in epithelial cells, in glandular cells, in adipocytes. And graded potentials are responsible for releasing neurotransmitters like acetylcholine.